We are now ready to begin to explore structures that are external to Norbert's cell wall. That is to say that although you can't see them on this model here, sometimes outside of this screen layer, there's a whole nother huge layer. And in fact, I thought JP might appreciate this the most. And I know there are others of you who are skiers as well, but you know the big puffy down jackets that we wear? Some bacteria have a big puffy down jacket that they wear. In fact, many times they have this layer of carbohydrate outside of their cell wall that is very protective, as you might guess. Additionally, just as Norbert does, another structure external to the cell wall is the bacterial flagellum, and we're going to learn about that and get to see how its motion is either clockwise or counterclockwise and how it moves like an awesome propeller. Thanks again to Isla and her mechanist abilities. Glycocalyx is a broad term to say that there's generally a carbohydrate, a polysaccharide layer outside of the cell wall that is very, very protective. Glycocalyces, that is plural, can take more than one form. So they can be either a capsule or a slime layer. And in both cases, they're very protective. In lab, we're going to talk in detail about capsules and their nature. Slime layers are a little less distinct than a capsule, but in both cases, they provide a lot of protection. For example, if a cell has a slime layer or a capsule, specifically, really, if it has a well-formed capsule, it has a very good defense against phagocytosis. And in this video, you're going to see an amoeba that is trying to engulf via phagocytosis two bacteria that are encapsulated, and it fails. So let's take a look. These two bacteria that it's trying to engulf, you can see the capsules around them. Notice there's kind of a halo around each of the two bacterial cells there. You can see that's the glycocalyx, and it's engulfed them, right? And it's going to try to take them in via phagocytosis, and it's kind of pulling on them, trying to bring them in. But as you're going to notice here, it fails, and no matter how hard it tries to phagocytose them, it's unable to do so. So it's allowed the glycocalyx on these two bacteria have allowed them to escape phagocytosis. Wow! Slime layers can have some of those same effects. It can also prevent hydrophobic toxins from getting in. Thomas will be interested to note that it can also protect from viruses binding, adhering, and being able to inject their nucleic acid. So there's a lot of protection afforded by capsules and slime layers. Another external layer that we sometimes see different than a glycocalyx is an S layer. The thing that makes an S layer different is twofold. One, it's the nature of the molecules making it up. That is, it tends to be protein rather than carb. So protein or glycoprotein, remember glycoproteins have carb regions on them, but we tend to see a polypeptide nature to an S layer. The other thing that differentiates an S layer is the composition, the organization. In an S layer, the glycoprotein or protein forms these very regularly repeating units, and it forms eventually what looks like floor tiles or ceiling tiles over the layer of the cell. So if you look really close at the ultrastructure, it'll look like this tiny little, very regularly repeating set of floor tiles. Sometimes they're even hexagonal. And what's cool about that is it's very, very regularly repeating. That leads to very succinct pore sizes in between the tiles in this outer protective layer. And so Ben and Sally are going to be super jazzed to know that this means that S layers can literally and have literally been used in biotechnology as membranes. Think of membranes with the smallest possible pore sizes and the most uniform possible pore sizes you can imagine. And in fact, if you go to the Web of Knowledge or the Web of Science, you're going to find a review article, a 2011 review article that's just entitled The Nanobiotechnology with S Layers. So S layers have a lot of application in biotechnology. But what do they do for the cell? Well, they're going to be very protective. They're going to protect against pH fluctuations, osmotic stress, enzymes. They're also going to protect against phagocytosis and toxins. So they have a lot of the same functions that a glycocalyx has. They also protect against predaceous bacteria. Later this semester, we're going to talk about a bacterium called Delovibrio. It just sounds scary, Delovibrio. Delovibrio is a predaceous bacterium that literally rams into bacterial cells. It actually moves them with its force, and then it starts rotating its flagellum, and it bores a hole into the outer membrane of gram-negative prey cells, and then it lodges itself in the periplasmic space, and it grows into a long worm-like structure and undergoes what's called multiple fission and breaks into many, many daughter cells, and it basically eats the prey cell alive. So that's what predaceous bacteria are all about. The other thing is that this may help the cell maintain its shape, rigidity, and promote adhesion. So we kind of recall that as being functions that we see of the cell wall, and so they're aided or aided and abetted by the S layer. Not all cells have an S layer. The most famous one that has an S layer is called Caulobacter crescens, and it's fun to look up and see some of the applications that has in biotechnology. Lastly, I want to talk about sheaths. Sheaths are another type of external structure, but rather than being a structure external to a single cell, they're a structure external to a population of cells. So we would have a whole bunch of cells housed by this outer sheath, and a sheath is kind of like a straw. You guys are totally going to laugh at me, but I wanted a demo, I wanted a model, and so a piece of one of the tubes that connects the gas to the outlet bit the dust, and I made a model. So you can see here, this is what a sheath would look like. So if each one of the beads inside of there are photosynthetic bacteria, such as cyanobacteria, and the outer tube is the sheath, see how this is enclosing a community. You've got inside of there enclosed a community of photosynthetic bacteria. And imagine how protective a sheath is to them. They've got this whole nice population cordoned away from predaceous bacteria and other threats. But also, this can hold on to surfaces. So, if my hand is a rock, here's this guy flapping in a stream or something like that. And hey, think about that. All the nutrients and food in the stream are flowing by this population of bacteria, and it's like fast food. They're like, woohoo, here comes my delivery food as it comes through on the water. So that's a pretty convenient thing. Let's summarize down in the picture below. Broadly, there are several layers external to the cell wall. One type are the glycocalyces that are polysaccharide, either slime layers, which are diffuse, or capsules, which are very, very discrete and regular. S layers made up of protein or glycoprotein, they're distinguished by their floor tile-like pattern. And finally, sheaths are a tube that holds a chain of cells together and gives them advantages in that chain. It's finally time to zoom in on Norbert's flagellum, and I want to make note that Norbert has a single flagellum at one end of the cell. We term that a polar flagellum. Additionally, I want to draw your attention to the parts of the flagellum. You'll notice that it has a region that's inserted into the cell wall. We call that the basal body. It then has this region that actually rotates. This is termed a hook. And then Isla's given this an incredibly beautiful filament. So you can see this long structure here is the filament. So flagella obviously involved in motility. They are sometimes single or sometimes multiple, but they're always these sort of hair-like protein structures. They are hollow cored. So in the long filament region of the flagellum, it actually is a repeating protein polymer that rolls around in a kind of helical way to leave a hollow core. So in fact, on this picture where we're really, really zoomed in to this particular cell wall, and a quick look at that will tell you whether it's gram-positive or gram-negative, right? So this flagellum emanating from this cell, and we can see that this is the filament. Further, this structure can be broken down into what's called the hook. And then lastly, this very basal structure that's embedded in the cell wall and membrane. And this is called together collectively the basal body. So hook filament and basal body comprise the flagellum. Sorry, O. And further, the basal body is constructed of all of these different rings. And the rings are named the L ring. So this top one there is the L ring. The middle one here is the P ring. And bottom down here are the, this one is called the MS ring. And this is called the C ring. They used to call these the S and M ring, and I think they got tired of me making fun of that and saying it's appropriate that it stabilizes a whip-like structure. No, I'm kidding. It had nothing to do with me. But nomenclature changes, right? So recognize these as sort of the structures that comprise the basal body. So it's interesting to note that next to flagella, there are some really lovely protein channels that enable the influx of the proton motive force, the PMF. And it's the PMF rushing in. So you've got a hydrogen ion gradient, right, in the periplasmic space of gram-negative cells that can respire. And it's the hydrogen ion gradient swooshing in to proteins that span the membrane that actually gives rise to the rotation of the flagellum. So as those protons rush in, you get either the clockwise or counterclockwise motion of the flagellum. So we might collectively say then that the PMF is the energy source that's used to spin the flagella. In either a clockwise or a counterclockwise motion. And they're fast. In fact, the bacterium Vibrio alginolyticus can actually spin, spell this right, can actually spin at 270 revolutions per second. This literally gives rise to a bacterium that can move at such a fast rate that if we relatively compared it to us, and we say, I was going to pick on James and say James is running across the ultimate frisbee field, and probably James, you're super fast, right? But if you think about the miles per hour that you could go, compared to this bacterium, you just pale in comparison. If you were as fast as Vibrio alginolyticus, you could actually run at 82 miles per hour, right? Nobody could compare, not even Usain Bolt. So not only are flagella rapidly moving, give rise to incredibly motile bacteria, but also sometimes they can serve as virulence factors. So we might say that they can be virulence factors. That literally means that they can contribute to the ability of a microorganism to cause disease. So I want to bring up our good friend Helicobacter pylori. I have an awesome stuffed microbe, one of my favorite stuffed microbes. And you can see that as I passed this around to my students one day, it acquired a gangsta headband. But in any case, Helicobacter pylori is, maybe it's a misunderstood gangster, because in fact it can cause peptic ulcers. We know that a part of what it does is contribute to the development or the disease presentation of peptic ulcers. And one of the ways that it does that is because it has what we call lophotrichous flagella. So check out this mohawk, but in fact these are a tuft of flagella at one end of the cell, lophotrichous. And these rotate, and when they do, they enable Helicobacter pylori to bore into the lining of the gut. Okay, so this leads to the symptoms of a peptic ulcer. However, as I said, misunderstood gangster, because in fact, if this is in the right balance in your body, it can prevent acid reflux. So it does good things too. It's just when it's in the wrong balance that it can present as disease. That being said, we should say a little bit more about that distribution of flagella, ranging from those that have a single polar flagellum, being monotrichous, versus those that have multiple flagella at one pole being one at each pole being amphitrichous, or multiple flagella at one pole being Lophotrichous. So different distribution, different names for the flagella. Unknown number 140 is a great example of something that has a polar flagellum, whereas Helicobacter pylori is a good example of something that has a Lophotrichous distribution of flagella. And then lastly, peritrichous all over the surface of the bacterial cell would be what we would see for E. coli if it's flagellated. Not all E. coli are motile, but if they are, they have peritrichous flagella. So now that we can see the distribution of flagella on the cell, let's talk about how the rotation, the propeller-like rotation of flagella will allow the bacteria to move in certain ways. That is, we know that they can rotate either clockwise or counterclockwise. And with that rotation comes certain types of movement. A counterclockwise rotation is going to be associated with the cell sort of moving forward, if you will, in a run-like pattern. We call it a run. That is a just straightforward movement. So when the bacterial flagella move in a counterclockwise way, we see them running. So we say that's a run. When they move in the opposite way, that is when they move in a clockwise direction, they sort of stop and reorient. We call that a tumble. So that's our stop, reorientation, that is a tumble. So this is moving directions or changing directions. So unlike James, when he's playing Ultimate Frisbee, and he can use what we call spatial sensing, where he can stand in one place on the field and he can see where the Frisbee is, he can move directly to that spot using spatial sensing. Bacteria would do no such thing. They use temporal sensing. That is, they sense one location that they're currently in, and in order to sense another location, they have to move. So bacteria have to constantly move in order to sense their environment and to tell whether it's changing. So the path that bacteria would take would be much more confounded than that James would. It would be more like a run, a tumble, a run, a tumble, a run, a tumble, a run, a tumble, and through a very convoluted path that we call a biased random walk, they would get to where they were going. So they don't use that straightforward spatial sensing. The process by which bacteria are moving in response to a chemical gradient is called chemotaxis. So while James is moving in response to where the Frisbee has moved, potentially, bacteria, they're often moving in response to food. Now, not to say that James doesn't do that too. Really, I know my skiers, sometimes I move in response to food, and that's true. Bacteria often are moving towards a chemical attractant, that is a nutrient, for example. And their motion is, as we said, very convoluted, because they'll sense that there's a gradient only by continuing to move. That is, they have to move in order to tell if where they are now is different than where they were a few seconds ago. So bacteria move towards a chemical or environmental attractant and away from a chemical or environmental repellent using chemotaxis and temporal sensing. That is, comparing the environment that they were in moments ago to the environment they are now in. So the chemical attractant or repellent enacts this change by binding to a transmembrane or integral membrane protein called a chemoreceptor. Once that chemoreceptor is bound up, it sends an internal signal that leads to the rotation either in a clockwise or counterclockwise way. So chemoreceptors affect the direction of rotation of the propeller-like flagella. So they control the movement of the flagella. In the absence of a chemical gradient, there are equal numbers of runs and tumbles. So what we see is right down here, when a bacterium is not moving towards some sort of nutrient, some sort of attractant, then it'll just move like this, very, very randomly. It'll move from one spot to another, to another, to another, with the hopes of sensing a difference between the spots, right? It has to keep moving in order to do that, because it can only use temporal sensing. So if it gets to one spot, say down here, where it's finally like, whoa, I sense something, then what will happen is that the number of runs the length of runs will start to get longer, and the number of tumbles will start to get less. So in the absence of a gradient, the bacterium has equal numbers of runs and tumbles, and it moves randomly. But if we put it in the presence of a chemical attractant, suddenly it's going to sense that, And it's going to begin to elongate its runs, more lengthy runs and less tumbles, until finally it takes this convoluted but a path that will get to the chemical attractant. So as compared to the random movement, we can see a biased random walk where eventually it gets from one low concentration in the gradient to the higher concentration of nutrient using this sort of movement. So this, again, is very convoluted, but it gets the bacterium there. And it's a dang good thing that the bacteria are wicked fast, because they have to take a very indirect route to get to wherever they're going. Certainly we know people like that too. Whenever they drive, they take the most convoluted paths. It takes a while for them to get there, but they get there. And certainly some of them drive fast enough that they might beat you anyway, right? So I'll give you a moment to jot that down. And maybe you're thinking of some questions that you might have regarding the movement of bacteria with regards to a chemical attractant. And maybe you're thinking, yeah, but they might move towards food, but don't they also move away from toxins or pollutants or waste? That is, as more and more byproducts of their metabolism accumulate, bacteria are going to try to move away from those byproducts, right? We do the same thing. The more we pollute areas on the earth, we move away from them and leave them for someone else to deal with. This is what bacteria do too, right? They'll move away from their own waste. It's really interesting to note that the relative sensitivity for an attractant is much greater than that for a repellent. In fact, there's double the number of chemoreceptors for attractants as there are for repellents. And the sensitivity for bacteria for attractants is much greater. That is, they will move with response to very, very low concentrations of chemical attractant, and they require much higher concentrations of repellent to move in response to those repellents. So the stronger force, and I guess this makes sense, right? The need to eat is stronger than the need to get away from repellents. If you go ahead and search YouTube for bacterial motility or bacterial runs and tumbles, you're going to come across a lot of really cool videos. One such video is done by UW-Madison bacteriology program, and I particularly like this image of Rhodobacter spheroides, and you can see how incredibly motile it is. Very cool depiction of perhaps the not direct but very rapid motion of that bacterium. Let's turn our attention back to Norbert and catch one more external structure. If you look at these sort of hair-like structures here, these are called pili or sometimes fimbriae, depends on the textbook that you're utilizing. So these structures are similar to flagella in that they are also comprised of proteins arranged helically with a hollow core, but they're much obviously smaller and thinner than flagella. Unlike flagella, they will sometimes work in adherence. So, for example, if you think about E. coli, the most common cause of urinary tract infections, it actually has an adhesin molecule on the surface of its pili that allows it to adhere to the urinary tract, and therefore it's not voided with the urine. It can hold on, and then that allows it to cause infection even in that area that tends to be cleared of bacteria via mechanical action. So they allow for adherence to specific surfaces, but they also will sometimes allow for movement of populations of cells on solid media. And this is really interesting. Those of you who have a passion for animals, and there were a number of you, will think this is so cool. There's a bacterium called Myxobacterium, and Myxobacterium can actually move as a population of cells in sort of this short twitching motility. And when these groups of cells all move together, it's literally termed a wolf pack. So a wolf pack of Myxobacteria moving by twitching motility. Now lastly, we should just briefly differentiate that certain surface pili are involved in conjugation. So remember, there are sex pili that have that specific distinguished function. Now, it depends upon the source you're looking at. Sometimes sex pili will be the only thing called a pili, and the others will be called fimbriae. Again, it just depends upon the lingo being used. We're going to stop right here, and thank you so much for your attention and your assistance in making the sort of blended portion of this class be really, really effective.